you're having a cell wall and the cell membrane. So if it is an isotonic solution, there won't be any net movement of particles, isn't it? So outer solution is same concentration as that of the cytoplasm. So here the concentration term is indicated towards surrounding environment and the cytoplasm of the cell. That's the most important point you have to understand over here. So the isotonic solution, the cell wall doesn't show any kind of interaction. But when you go for hypertonic and hypotonic, there's a small mistake over here, hypertonic. Okay. So when you take the hypertonic solution, what happens? So that's hypo. Clear? Now when it is hypo, what happens? Movement from inside to outside happens. Because of the hypo and hypertonic differences, there may be shrinkage called plasmolysis or swelling called lysis. All these are happening in case of animal cell. But in case of plant cell, it won't happen because the cell wall will help in maintaining the pressure which will be put on the cell because of the outside concentration changes. The rigid cell wall. That's the important point over here. How it is tolerating? By maintaining a hydrostatic pressure inside the cell. That's what I showed. The cell around which you will be having a cell membrane and cell wall. So if a pressure is put on the cell, it will be counteracted by outside pressure. So that's how a plant cell wall maintains the turgidity even though the outside concentration changes. Okay. So the same points which I explained in the previous slide. Prevents the cell swelling, 1 point. And it balances between the cytosol and extracellular fluids. Cytosol is nothing but the fluid inside the cell, and extracellular fluids is nothing but the outside the cell fluids. In between, inside the cell and outside the cell, there will be a balance because of the presence of cell wall. So the plant cell will not burst out even if you keep it in a high concentrated solution, unlike an animal cell. Okay, then that's all about cell wall. Then we get into the plasma membrane structure. When you take the plasma membrane, the plasma membrane structure exactly was given by a scientist, Singer and Nicholson, in 1987. Membrane structure, but it was accepted after 10 years. Why it took those many years? I'll explain while explaining the structural aspect. Okay. Now, when you see the cell membrane of a plant cell or an animal cell, both cells will have the. It was accepted in 1972, but is proposed in long before that. Okay, so you will find in animal cell and even in plant cell. In both, you will have the same structure. So the Singer and Nicholson are the scientists. The structure name is fluid mosaic. Why the name fluid mosaic? If you take the cell membrane structure of any cell, you will be having lipids, you will be having proteins and carbohydrates also. All the biomolecules you will find in a cell membrane. Let's see how each individual biomolecule enhances the structural and functional aspect of cell membrane. When you take lipid, here the lipid exists in the form of a bilayer. You'll find it 2 layers. This is 1 layer and this is 2nd layer. So these 2 layers were called phospholipid bilayer. Phosphate group will be attached to a lipid molecule. This attachment is quite special. If you take this as the lipid molecule and this as the phosphate molecule, this is called head and this is a tail part. So if you observe, the head part is hydrophilic. Phili means attractive. Attractive towards water. Is there at the outer surface and at the inner surface. Observe, you are having 2 layers. The first layer, hydrophilic head, is outside, The second layer, hydrophilic head is inside. So both the hydrophilic heads are outside and inside of the membrane. Whereas the tile is hydrophobic. Phobic means repellent. It will repel water, which is there inside. See, observe the diagram. So when you take lipid exists in the form of bilayer and in the form of phospholipid nature. When you take the phosphate and lipid nature, the hydrophilic heads and hydrophobic tails are arranged in the form of two layers. That's the first part about lipids. When you take proteins, proteins may be either embedded inside the plasma membrane like this, the purple one, which are called integral proteins, or the proteins may be at the surface of the plasma membrane like this. So those are called surface proteins. Then when you come to carbohydrate part, the carbohydrate may be associated with lipid or may be associated with protein. Here, if a carbohydrate is associated with lipid, it is called glycolipid. If the carbohydrate is associated with protein, it is called glycoprotein. See, when you take the whole plasma membrane structure, you will be having outside of the plasma membrane and inside of the plasma membrane. Okay, so this is the outer part and this is the inner part. So it was clearly written in this diagram: external surface, internal surface. And in between the external and internal surface, you will be having several molecules like the phospholipid, which is having hydrophobic and hydrophilic nature material. Then the proteins, you are having proteins which are there inside, spanning between the plasma membrane, integral they are called, and some proteins which are there at the surface only, called peripheral proteins. Okay, peripheral nothing but a superficial surface. And then you are having carbohydrate chain. The carbohydrate may be associated with lipid, glycolipid. If it is associated with protein, glycoprotein. So this is all about the fluid mosaic model, where you will have a bilayer in which lipids and proteins are embedded. Just a simple structure. But what I'm very much interested to discuss is why the structure is like this. Why this structure was accepted universally. So that I'm going to explain using two functional aspects of plasma membrane. Other name for plasma membrane is plasma lemma. So this both are same words. Okay. When you take plasma membrane, the plasma membrane structure is so different that it protects both plant and animal cell. Previously, the plasma membrane structure was proposed in the form of unit membrane. Actually, if you understand this, the next concept will be much easier. So it was explained in the form of unit membrane. What does mean by unit membrane? Nothing but this theory explains that the structure of plasma membrane is having molecules like lipids and proteins. Lipids and proteins will be there in plasma membrane structure, which acts as gateway. Gateway means what? They will allow only few molecules inside the cell, and they will allow only few molecules to go out of the cell. So this inflow of molecules into the cell and outflow of the molecules out of the cell are determined by the structure of the plasma membrane. So the unit membrane says that the plasma membrane is made up of lipid molecules and protein molecules, which acts as gateway and allows only few molecules to get in and get out of the cell. This was the theory proposed at first. But in later years, so many studies reveal that the plasma membrane is not only made up of lipids and proteins, even some other molecules are also involved in plasma membrane. So how the different molecules are involved and how they are arranged in plasma membrane structure was explained by Singer and Nicholson. These are the scientists' names. Their theory was named as fluid mosaic model of plasma membrane structure. Fluid mosaic. Now we'll start from here. What does mean by fluid mosaic? Other than the unit membrane structure, the fluid mosaic model explains some more points. When you take the fluid matter, so here the fluid matter is nothing but the proteins and the lipids matter which makes up the plasma membrane. Within this proteins and lipids fluid, there is a mosaic. Mosaic means what? A zigzag way. The tiles arrangement on floor, we call it mosaic, isn't it? So the mosaic manner of arrangement of carbohydrates is observed in plasma membrane. This is all about fluid mosaic model of plasma membrane proposed by Singer and Nicholson scientists and was accepted in 1972 year. Now when you take under electron microscope, the study under electron microscope reveals structure like this about the cell membrane. Cell membrane is another name for plasma membrane. Simply plasma membrane, plasma lemma, cell membrane, all are synonymous words. Now when you take under electron microscope, the plasma membrane shows various structures. It something looks like this. When you observe the whole structure, you're having an outermost part and inside some structures are there and innermost part. Okay, so here innermost means what? This, if I assume that this is the cell, so here this is the plasma membrane around the cell. So that is innermost part. Outermost part means what? Assume that this is the whole plasma membrane. This is the extracellular side, outside of the cell. Okay. Now when you take the whole plasma membrane, this as a part like this, you will be having first a bilayer. Bilayer. Assume that here you are having one layer here, one layer here, dividing each part into two layers. Okay. Now, each layer if you take in detail, this is the part of one layer. I'm circling in the diagram. Observe it. This is the part of one layer. Okay. And this is the part of second layer. So that's how you got bilayer structure of plasma membrane. Now, what are there in this bilayer? If you take in this bilayer, one layer individually, I put a block on there. See, you're having a molecule with a head part and a tail part. Okay, something rough diagram looks like a puppet. Okay, so head part and a tail part. And when you take the head part is called hydrophilic. Hydrophilic stands for hydro, water, philic, attractant. So the head part, which is shown in this layer, all the blue ball-like head parts, they will attract water. Here, then you are having a tail part for each blue color head, isn't it? So I'm just drawing again on the diagram. See, one layer I'm explaining. So the tail part, if you take, the tail part is hydro, again water, phobic, repellent. It won't like water. What happens? The tail part will not absorb water. So it will not absorb water, whereas the head part will absorb water. So such layer, two times you are having. See, this is one layer and this is second layer. Same structure. The whole glass membrane is made up of such two layers. Observe, I am circling on the diagram. One layer, two layer, bilayer. One layer, two layer, bilayer. Okay. So the bilayer will have a head part and tail part in each layer. Now what is this bilayer? The bilayer is made up of phospholipid. Okay. So here the head part is the lipid molecule which attracts water. That's why it is hydrophilic. And the tail part is the phosphate group which repels water. Okay. So that's why it is hydrophobic. So if you take the single layer, just observe the diagram, the outermost ball parts will take up water. And the innermost ball parts, they also will take up water. But in between you are having the tails, no? They will not take in water. So this difference shows what molecules will get in, what molecules will go out. This nature we call as selectivity. Isn't it? If you are choosing something, what we call it? We call it selectivity. So that selectivity is shown by phospholipid bilayer. Just repeating, phospholipid bilayer. You will be having two layers in the cell membrane. What are those two layers? Lipid molecule and phosphate molecule making a head and tail of the layer. When you take the head part, they are hydrophilic. Understand the word? Hydrophilic means they will take up water molecules. Whereas the tail part you take, they are hydrophobic. They will not take up water molecules. They will repel the water molecules. When you take the lipid molecule, this is how the phospholipid layer will be found in the form of bilayer. That's one part of the plasma membrane or cell membrane structure. This is one part. Over this part. Now coming to the second part. You'll be having various proteins also in the cell membrane. The proteins, if you observe, they may be existing in different forms. See here, in the diagram I'm showing integral protein. So integral means this purple one. See the purple one? How they are present? They are present from outside to inside. They are spanning the whole bilayer. Isn't it? If you take this is the whole layer of the plasma membrane, it is spanning the whole layer. It's passing from the outer layer towards the inner layer. Isn't it? It is shading the purple ones. So those proteins we call as integral. Integral snapping part which can pass through the plasma membrane. Those are one kind of proteins. You're having second kind of proteins which will be there only on the surface. They'll not pass through the plasma membrane. See here, again purple color protein molecules are there. They're just on the surface of the bilayer. Observe it. I'm shading on the diagram. So those are called surface proteins. So you are having proteins in two types within the cell membrane structure. One protein which will pass through the plasma membrane like this. We call them as protein channel. I'm circling on the diagram labeled part. Protein channel means what? Some of the molecules will come inside the plasma membrane. No? They'll pass through this like this. Okay. So through the protein channels which are integral proteins, molecules will move into the cell. Or molecules may move out of the cell. So this inflow and outflow is also controlled by proteins. So phosphobilayer also controls movement protein and the proteins also controls the movement. So both these are, we can say, for selectivity. The same word, choosing the molecules moving in and out of the cell membrane. Now the proteins may be throughout the cell membrane like this, or they may at the surface of the cell membrane. Based on that, we are giving the terminology. integral proteins and surface proteins.